Now, I have the pleasure of introducing our next two speakers. First, Dr. Erin Hancock. Erin received her PhD in organic chemistry from Indiana University and BA in chemistry from Hanover College in Indiana. Currently, Erin is a research investigator at Corteva AgriScience and works as a discovery chemist in a variety of therapeutic areas. Her primary focus is on leveraging innate activity and sustainable attributes from natural products, and she also contributes to late-stage discovery, fungicide, and insecticide optimization projects. And second, we have Ashley Geraets. Ashley joined Elsevier in 2021 as a customer consultant. She is responsible for our chemistry solutions portfolio in North America and helps corporate end users get the most out of Reaxys through product trainings and consultations. Before joining Elsevier, Ashley earned her PhD in chemistry and master's in chemistry with a focus in forensic biochem from the University of Central Florida and her BA in biochemistry from Washington and Jefferson College in Pennsylvania. Welcome, Erin, Ashley. The stage is yours.
All right. Well, thank you, Randy, for that introduction. And on behalf of the organizers of Reaxys User Day and Elsevier, I'm really excited to have some time today to hopefully teach you a thing or two about Corteva AgriScience and the work we do here with regards to developing new molecules for crop protection, and specifically how we leverage Reaxys within our active generation teams. So just an obligatory agenda for the presentation. I'll start with a quick bio and then introduce Corteva AgriScience and how we think about crop protection discovery, and then move into the implementation of a variety of features from Reaxys to support some of our active generation teams. So a bit about myself, in addition to what Randy had provided, by my training and passion, I'm a natural product chemist at heart. I've studied quite a bit in Indiana, which is in the United States, a few hours south of Chicago. And so my PhD focused on organic chemistry with Kevin Brown at Indiana University. And there I spent time synthesizing two members of a laterine family of natural products. And if you haven't seen these molecules or heard of them, they're quite interesting from their structural complexity. There are 3 to 5 iterative cyclobutanes. So if you think about the structures in 3D, they do sit like a staircase. And then moving on from completion of my PhD in 2020, I have been at Corteva AgriScience for about 4 years now. And like Randy said, I've had the opportunity to work on a variety of therapeutic areas: fungicide, insecticide, herbicide chemistry, and throughout various stages of the pipeline. And what I'll focus on today is our active gen teams and some natural product chemistry. Switching gears to Corteva AgriScience. Corteva AgriScience is technically a fairly new company. We just celebrated our 5-year anniversary last week. But the company was founded on the rich heritages of Dow AgriScience and DuPont Chemistry. And so these two companies merged and started back in 2015, and their agricultural sectors spun off, and that is how Corteva came about. So with that, Corteva AgriScience is still a fairly large company. We have about 20,000 employees worldwide, spanning over 130 different countries. And within those 130 countries, we have over 140 R&D facilities, and all are working towards the goal to enrich the lives of those who produce and consume, ensuring progress for future generations to come. So growing up, my family, we do not have background in farming. Growing up in the Midwest, of course, I drove by cornfields and soy fields quite frequently, but it wasn't until I came across Corteva AgriScience that I really began to understand the benefits of crop protection. So here in this handful of photos, you can see the damage that can occur to farmers in their fields if we aren't able to control weeds, if we can't manage diseases such as a different fungi, if we can't control pests such as chewing insects, caterpillars, nematodes, for example, and if our plants don't get the proper nutrients, then yields will definitely decrease, affecting communities that the farmers work with. So ultimately, the goal of crop protection solutions is to take fields that look like the images shown above and transform them into high-yielding and sustainable food sources for their communities. Now, how do we do that specifically? Like many companies, we also have a pipeline that is similar in the sense that it's a giant funnel. So early discovery stages, such as our hit and active generation indicated by the blue section, work on identifying and inventing new molecules that have activity to kill the bugs, weeds, or fungi. And as it moves down, the amounts of molecules we hone in on will go through a series of optimization until it reaches our discovery process or process and science technology, where they'll further improve the route, longer term safety studies, etc., until we come to launch. And again, the talk today will focus on the blue section, the hit and active generation, in our discovery team. So zooming in a bit more on the active gen, we have three groups or three sources of inspiration where we try to identify new biological activity. So our first is natural products and biologicals. And as the name would describe, this group focuses on total synthesis, semi-synthetic efforts, fermentation, etc. Bioactive hypotheses find inspiration from literature, other things like targeted acquisitions, retrospective searches, as well as looking at some other computational methods such as reverse stocking. And last but not least, the competitor-inspired group, as the name suggests as well, looks at competitor patents for scaffold hops, etc. But all three of these groups are tagged with the responsibility to identify reproducible biological activity and ensuring that there's synthetic opportunity with the molecules. Now, like many research programs, we do have a design, build, and test approach to analog sorting, design, and optimization. And so as we start to see promising hints of potency, spectrum, and we do establish synthetic direction, these molecules will continue to move through the pipeline until ultimately a product is launched. I also do want to highlight our testing cascade and biological assays, as I think they're quite unique. We do have the ability to go in vitro or enzyme assays, and we have the capabilities to develop those assays based off of the mode of action or hypothesis we are looking at. But if the opportunity presents itself, we go directly to the plant, the insect, or the fungi. So the images on the bottom of the screen are our weed management or herbicide assays. So our level 1 test, we start with a high throughput screening with 2 different weed species, a grass and a broadleaf. And then once we see activity, we move towards more robust greenhouse tests. You can see the plants are larger. So we're working with more of the plant mechanisms, metabolisms, etc. Translation then proceeds to field trials. And now field trials are quite interesting because we see parameters such as UV stability, rain fastness, etc., to really test the chemistry we've developed. And in all doing so, sustainability is important to us. So the image on the right is our apiary or beekeepers on site. We do both in vivo and in vitro testing to ensure the safety of bee populations, as well as other ecotox, mamtox, and screening of physical properties too. So now let's get into how we've implemented features from Reaxys to support the ActiveGen teams. So there are 3 areas that I want to discuss. One is how we manage our IP and understand the prior art around areas we're pursuing. The second is from a synthetic standpoint, so leveraging Reaxys predictive retrosynthesis. And then last but not least, highlight our natural products platform and some of the AI implementations we are using. So with regards to intellectual property, as we discover, develop a molecule, we want to explore the SAR, or the structure-activity relationship. And before we can pursue those, we need to do a thorough investigation to make sure we're not infringing on any other patents or be aware of prior art. So the molecule on the left, UK2A, it's actually a natural product, and we've launched several different fungicide products from this molecule and provided it as a source of inspiration. So you can imagine, as we're looking to understand what IP space we're operating in, you can start to manipulate several different portions of the molecule, as I have abbreviated with R1, R2, Q, etc. And so if there's about 7 variables, each maybe 4 to 7 different identities, you can then imagine that there are a lot of different possibilities. And Reaxys is great because we can run these Marcouche structures, and we've done over 3,500 variables. Secondly, we use Reaxys to broaden our quick search. So if you do just a traditional search, there are a few features I'd like to highlight that allow you to explore additional molecules other than the ones that you have drawn. So first and foremost, our Marcouche structure is in the blue dashed box, and you can use the smart R groups indicated on the left to further expand your R group identities. And what also is very nice is that you can, if you look at R1, instead of having just a normal pyridine ring, you can use the any atom feature, and it will include any atom, as you can see indicated by the A on the arrow group on the right. And then last but not least, the Reaxys generic groups are very helpful because you can broadly include things just like alkyl, aryl, or heteroaryl, and they have a vast array of options there. And the third thing is that, we can identify similar or related compounds through similarity or substructure searches. So this top screen or top box is the structure as drawn, and you can see we only identify 70 compounds, but if you were to broaden it to any substructure or similarity, on the bottom you can see that we're now expanding our search to include 245, or if we do the widest similarity, over 1,000. And the last thing I want to highlight, we also use the query builder quite frequently. The query builder is really nice because it comes with preset forms to where you can plug in the information necessary for your search. So in this case, I have the natural products form where I've inputted the structure. You can specify what organism you are looking for this to be isolated from if you know that. The second form I have is the patent form. You can add things like patent assignee or inventors. In this case, I put a publication year to look for anything of relevance or still within the patent life. And then last but not least, I added a biological species field. I mentioned earlier that this molecule is a fungicide and does target Asian soybean rust. So then you can put the organism of interest there. And ultimately, this again allows for a very thorough search. And what we do like a lot about this is we can export and save and share searches so that we can continue to build off of them as we expand our IP. Moving on to the retrosynthesis, predictive retrosynthesis. This is something that I use and the team uses quite often. It's a great catalyst for ideation. So as you learn more about an area, about your SAR, you begin asking questions like, what happens if I remove an oxygen? Or in this case, maybe the carbonyls highlighted in the orange boxes. And these searches, what they provide are both predicted and published reaction data. Additionally, they perform searches autonomously, which is fantastic because you can, look at several different structures, several different parameters with a search, and come back within an hour or two and then review the results. This is great for de novo synthesis or scaffold hops, as well as not only does it just provide published routes for compounds, but it will provide similar compounds that are published. So perhaps that can be very insightful. Here's an example of one of the searches I did where if we were looking at removing the 2 carbonyl groups for just CH2s, you can see, and I think Ashley is going to demo this next, but you can toggle on and off predicted versus published routes. And once you move into the edit window, you can change the number of iterations a search does, for example, starting material searches, etc. But as the algorithm develops predicted routes, it'll then pop up on, as you can see on the bottom image, where this search did come up with 4 predicted routes. And while it did not have any published routes, we can see that there were some related compounds that it pulled literature references for. Once the search is ran, here's an example of what the window will look like. The top bar with the green boxes are the tree views of the different routes. So this is quite a complex molecule, and the routes are quite long, but if you shorten the routes, you'll start to be able to see a more divergent or convergent pathway depending on the synthesis. The predicted route itself has several points of tunability. For example, it does trace the starting material back to commercial sources, but for instance, if the material is too expensive, or perhaps you want to prepare it in-house, you can have it to further trace back the material to new commercially available building blocks. And then if you hover over each reaction step, there's 3 dots to where you can select different customization parameters. You can delete steps, you can add or remove, for example, which can help you fine-tune your synthesis. And then one of the things I really like about thinking about things retrosynthetically is often each step is kind of grouped by the disconnection. So then you have the known literature examples at your disposal versus having to do several forward searches consecutively. So even though this first example is just a benzyl D protection, you can see a familiar search result where the experimental procedure can be toggled on and off. You have the reference, the yield, and the score on the right is like a feasibility score. So that also helps you rank the feasibility and similarity of the known literature examples. So this brings me to the final section of my talk, and that is our natural product discovery platform at Corteva. So traditionally, when you think about discovering new natural products, it's more of an imperial screening strategy where oftentimes it's geographically relevant. You're looking for biodiversity, bioactivity via guided isolation. And what we've been focusing on recently is creating an integrated and information-driven platform which leverages targeted isolation from genomics and metabolomics. And so with that, Corteva, we have over 600,000 cultures, so quite a diverse collection of different strains from both bacteria and fungi. And these strains are then subjected to our high throughput fermentation, to where then a variety of analytical techniques and bioinformatics are used to help us identify metabolites or microbes of interest. From here, these isolated metabolites or just series of microbes are then subjected to our traditional crop protection assays, like I had shown earlier in the presentation. Once we have a hit or we do see some activity that's exciting, we then have a variety of avenues to where we can further investigate the metabolite or microbes. So first are our synthetic efforts. So we do have our discovery chemistry synthetic teams, again, targeting de novo synthesis or natural product mimics. And then we have semi-synthetic products too, which can be accessed through fermentation and then maybe one or two synthetic steps afterwards. We have bioprocess, biofermentation, optimization, and scale-up to where we can isolate pure natural products, or even strain optimization, which then provides live microbes, and that's part of our newer biological sector. So again, with the theme of how AI has impacted our discovery, is that, of course, as I've mentioned before with Reaxys, our chemical synthesis team does rely on Reaxys from a predictive synthesis standpoint to help us explore new areas of chemistry to really be that catalyst for ideation and route scoping, as well as traditional conditions, etc. Apart from Reaxys, though, we have implemented AI to help us create molecular networks through identification of a variety of different chemical similarity between groups. We've also been able to do structure prediction with MS2 data, predict gene clusters and biosynthetic pathways for metabolites we identify. And I'm sure most are familiar with AlphaFold, protein folding prediction software. And so with that, hopefully I've been able to give you a glimpse into how Corteva thinks about developing and inventing new crop protection molecules, and how the reproducible biological activity, synthetic opportunity, and sustainability are key drivers. We have lots of tools and capabilities to assist in this process, and in particular today, I highlighted Reaxys, and whether or not that was from a IP standpoint or helping teams develop robust reaction routes, it does provide a means to explore these areas. And last but not least, I would like to take a few minutes to thank the Reaxys User Day Organizing Committee, Elsevier, Ashley, Eleonora, and Sonam for their help with the presentation. And Celso, he's one who's at Corteva who does a lot of these IP searches for us and really help project teams out, as well as our active gen teams highlighted today. And of course, thank you all for your time and attention. I'll pass it over to Ashley.
Erin, thank you so much for highlighting how you are using Reaxys at Corteva. So I'm going to quickly share my screen and elaborate a bit on some of the information that Erin shared during her talk. And so for those of you that have seen Reaxys before, this is our landing page, and I'm actually going to go straight into talking about Reaxys predictive retrosynthesis. So at the top here, I'm just going to select our retrosynthetic tool. At Reaxys, we know that a lot of chemists like Erin are looking at how to make something and even what to make in order to move forward their processes. And so when we take a look at Reaxys predictive retrosynthesis, we are looking at how do we design a synthetic route quickly and execute the most efficient synthetic route. And there's a few ways that we can do that. And you, as a user of predictive retrosynthesis, could make it fit your success in the synthetic pathways that you're looking at. And so, as Erin did highlight, there is a predictive portion of the tool and a published portion of the tool. When we look at the predictive portion of the tool, we are able to do some fine tuning that may assist you as you use it. You can edit the length of the route that you may be interested in. You can set it to last step only. If you're really looking at idea generation, you're also able to change the diversity of the routes or implement that. There's also some standard and extended synthesis time. And then, of course, in Reaxys predictive retrosynthesis with pending AI, you're able to select some building blocks depending on what you're most interested in. So we really wanted to give chemists the ability to ideate quickly, really look around and unlock their synthetic challenges and create routes that are going to be efficient for their teams. And so go ahead and close that, and I'll leave both the predicted and the published routes on, but I really want to dive into showing how you can use the drawing tool to get started and then actually highlight the individual steps of the process. And so in Marvin.js, which is the drawing editor we're using, I'm able to either draw a structure as Erin highlighted with the Reaxys group generics, or any of the tools on the sides here. You can also instruct a structure from names. You can do that up here. I'm actually just going to import a structure so I don't have to draw anything. Choose a file and I'll bring that in really quick. And I just selected a structure that may be of interest for individuals, but of course you could bring in any structure that you're looking for here. And then if I was interested in selecting a specific place on the molecule that I really wanted to protect or at a breaking point, I can do that here as well, but I'm not going to do that for today, but just wanted to highlight that those features are also available within the predicted tool. I'll go ahead and select synthesize, and it will begin the run. Once you've started your run, you'll see that I've ran this one in the past just to make the process here a little bit quicker, but it will begin in the queue and it will take some time, but you'll be able to have it saved here. You can see I have several different structures saved in this process, but we'll go ahead and look at the structure here. You can see we have 12 predicted routes, 0 published routes, and we'll be able to view. If you did happen to change or want to take a look at your parameters, you can select this arrow here, and then it will show the parameters in which you're working with inside of the route running. Go ahead and select view. And this will look similar to what you saw when Erin was doing her talk. You'll be able to kind of see the individual pieces of the route, and you'll be able to take a look at all of the information. So your target will be shown at the top. You'll also be able to view the number of steps, the route topology, that score, and then go directly to your route. At this first step, you're able to take a look at all of the routes that were a part of the result set, and you can take a look depending on if you'd be interested in kind of something more simple. But if you're also able or would like to take a look at something with more information, you can do that as well. And so I'm just going to select one of these routes. We can take a look. I'm going to go with predicted route 6, select the tree view, and it's going to bring us into our overall search results. Now you are able to see all of the different routes that were created. So you could jump through them to take a look at different ones. So if this doesn't really fit what you're interested in, you may
Thank you, Erin, Ashley. Those were great presentations. So we do have several great questions in the chat. I'll start at the top. Erin, how does Corteva mitigate off-target effects of herbicides, fungicides, pesticides at an early stage? And then also, are there similarities to drug discovery approaches?
So the first part of that question was about mitigating off-target effects early stage. So we have several ways we do that. We have a lot of known modes of action that are undesirable, whether or not it's from there's known resistance with that site, for example, or just, from past learnings. We run all of our compounds through those, and if they do flag those assays, we either try to learn more about the SAR, and if there's other analogs we can pursue that don't have that off-target effect, or sometimes we just have to make the decision to leave that area. Other ways we do that too, we will run a variety of other biological assays, so maybe for insecticides, for example, to look at the symptomology or that they display when subjected to different AIs, and some of those responses too can guide us to mitigate any of the undesirable target effects. The second part of the question, similarities to drug discovery. There's a lot of overlap, I think, between pharmaceuticals and agriscience. Both companies, both areas are making really complex molecules to solve some very difficult problems, and often, similar computational modeling's used, reverse docking, harnessing if the crystal structure is known or if the DNA sequence has been isolated. All of those tools we use as well to answer very similar questions.
Thanks, Erin. You mentioned SAR, so there's another question around that topic. When analyzing SAR to identify areas that are IP protected, how are the initial scaffolds identified? And then the second part of that question is, where do you obtain the bioactivity data used for SAR analysis?
Yeah, so the first part of the question with regards to identifying areas that are protected by IP and how those scaffolds are identified. Sometimes those scaffolds come directly from competitor patents, so we know the IP around those by reading the claims, etc. Other times, if we find an interesting molecule, we'll use Reaxys as well as other search platforms to make sure we do a thorough search to see what related analogs are known from an IP perspective. And then from there, make an assessment if it's open or operable, and continue from there. The second part was how we generate the bioactivity. So we have greenhouses on site that we can look at, crop safety as well as growing a variety of different weeds to test herbicides on. We have an insectary where we have colonies of, chewing insects, sap-feeding insects, as well as we have a vast collection of spores to inoculate the plants with a variety of different fungi, and then see how the molecules behave or how efficacious they are. And from there, we can compile the biodata and incorporate it with the structure to develop SAR.
Thanks, Erin. Now, a third question. This one has two parts. What other ways have AI or AI tools impacted lead generation and small molecule discovery? Second part is, how do you see these tools impacting future workflows?
Yeah, so apart from, with regards to the tools AI tools we're using, apart from, retrosynthetic planning, a lot of the AI is focused on our computational, developing predictive molecules to incorporate structure and bioactivity is one area we're looking at, as well as, I think some of this is pretty cool. It's not maybe directly towards SAR, but we do have seed apply technology and traits division as well. And they're using AI to help sort seeds and identify if they're formulated properly. We also use AI to help, grade biological tests. For example, with insects, a lot of times it's very manual. You have to count how many dead bugs are on the plate. But now with AI and imaging, we can, feed the algorithm pictures, true, false scenarios, and then it can perform those tasks versus our scientists. And I think future-wise, as everyone in science, we're constantly learning and incorporating new technology as it comes online, where we've partnered with a variety of companies and academic institutions to help us leverage learnings that are coming up within the space so we can stay on the cutting edge of science as well.
That's a super exciting field. Thank you, Erin. We have several more questions. I know I'm peppering you with quite a few, but there were a lot of interest in the chat. So the next question it's 3 parts. How often are prior art queries performed? Is that by project or ongoing? What would be your recommendation?
Oh, great question. So how often are queries performed? They're performed quite frequently, especially so at the start of a new area. It's always good to perform those searches so you can understand what's been claimed, what's available. You definitely don't want to infringe on any patents or IP. And then every time you start to change the molecule, change the core, or find a new pharmacophore or modify one, it's always good to go back and rerun those searches because those initial Marcouche structures, while they are general, patent attorneys and folks have gone really good at justifying their language to include, one side of the spectrum versus the other side. So it's always recommended to repeat those queries every time you begin to deviate from your initial search. And so, with that, my recommendation would, to repeat those searches regularly, and especially when you deviate from your initial molecule.
Thanks, Erin. So another question. First of all, the question is starting with a thank you for the great presentation. Regarding the final part of your presentation on MP discovery, how do you identify key proteins to be targeted? Additionally, how do you integrate internal bioactivity data with data from the literature?
Okay, that's a really good question too. So with regards to identifying proteins, I'm assuming that's maybe thinking more of like enzyme or the active site. So we will use literature if it's available, but one thing that I learned quickly when I joined was that there's far less known about the proteins and active sites in plants compared to bacterial or other fungal systems that are drug targets for human health. So a lot of times we will, take information from the literature from that and try to develop homology models within a plant species we're looking at. We have very talented biochemists that are able to maybe overexpress and isolate proteins that we can then do subsequent studies on as well. And our computational department are also very talented and can help, predict while maybe the biologist is working on the isolation, and then the two can work together to provide more information for that.
Erin and Ashley, thank you again for your presentations. Erin, thank you for answering all these great questions. Moving on, we have a scheduled break until 12:35 Eastern time. However, if you'd like to stick by your screen and enjoy your lunch, or if you're on the West Coast, enjoy your coffee, I'll explain a little bit more of what we have planned during this break. Thank you again, everybody.